So it's often surprising that we can learn about aging by studying flies and worms and even yeast cells.
You know, in biology, when you've got complex problems, the place you look for answers are simple animals.
It's underappreciated how closely related we are to mice, and it's even harder to imagine that we're related to even a simple yeast cell. But it turns out that we're quite closely related to them genetically, and even in how we live our lives.
They grow, they develop sexually, they interact with their environment, they sense things, but eventually things start to slow down and they stop reproducing, eventually they stop eating, and they die.
The buck actually has studied a large number of simple organisms such as Caenorhabditis elegans. This is a small worm. It's about the size of a comma. It is very simple. It lives 20 days.
There's incredible power there because you can do lots and lots of experiments really quickly when you've got an animal that's aging that rapidly.
If we look at the example of Drosophila, so this is the fruit fly. If you look at their genomes, many of the pathways that control aging in Drosophila are actually conserved in humans. If the fruit fly lives 70 days and a human lives 70 years, well, you've accelerated the speed at which you're working by 360-fold.
That's actually why we made rapid progress in the beginning. We were able to finish the experiments very quickly and move on and find more genes that were controlling aging.
They allow us to start asking very quick questions and to answer them.
It's really been a revolution using these model organisms because we now understand a lot of the fundamental processes of aging itself.
Many of the findings that have been made in these simple organisms can now be translated into humans.